right? So as Corinne just did. So if you want to remain off camera, that's totally fine. So how this will work is that Susan will give her presentation about 15-20 minutes, and you can put your questions in the chat in the meantime, or afterwards we'll just have open Q&A session afterwards. Sorry, we're done then. Anyways, so we have Susan Brawley, who is a semi-retired professor from University of Maine, a biology professor, and she has an action-packed presentation. So go ahead, take it away. Oh, and if you could also please, while she's presenting, remain on mute. Thank you so much. So go ahead, Susan, take it away.
Okay. So here I am standing in a rockweed bed at Schoodic not long ago, and you can see I'm holding up an Ascophyllum, and it forms an air bladder every year after the first one. And so you can determine the minimum age for a rockweed by counting the air bladders. As I don't remember doing that to that one, but it looks like it was 5 or 6 years old. So what I'm going to do is talk about where and what is Ascophyllum nodosum, its ecological importance, and some uses of Ascophyllum. And it'll probably be 20 minutes, and then we've got 40 minutes for people's comments, questions, discussion. So isn't this a beautiful bed of Ascophyllum nodosum? This is at the edge of Winter Harbor a few weeks ago. And I realize I need to move you guys so that I can see. Okay, so this is just a few weeks ago at Winter Harbor. And as you can tell, Ascophyllum is incredibly abundant. It's especially abundant in calmer shores. On the very outer coast, it breaks too easily to be abundant like this there. And it forms a canopy that protects other organisms in the mid-intertidal zone from high temperatures, high light, and drying. The canopy in the lower area has been found to sometimes increase predation on other organisms by green crabs, which are a non-indigenous species. And the non-indigenous periwinkle snail is very common there too. So Ascophyllum individuals form receptacles, and these are the receptacles. And because this is a herbarium page, they were pressed, and all of these sperm, which are coming out of conceptacles in the receptacle, you can tell someone wanted to torture students when they did this naming. All of these orange splotches are full of sperm. So there are male individuals, and then there are female individuals, and the eggs would be green if we had a similar blot. Now, these are obviously fully mature receptacles, and this was collected in April. Here we have another Ascophyllum, which came from the Darling Center Shore, our marine lab. And there are very small, immature receptacles forming because this was in September, in fact, of 2021. And here's the holdfast. And these are what are called base shoots. Typically, they are suppressed under the canopy of the Ascophyllum until part of it is broken by storms, or this one would be too small to be harvested, but at any rate, anything that causes more light to come in will start to cause those shoots to grow up. The receptacles release their gametes in spring. And here we're on the outer coast. Okay. We're on the outer coast at Schoodic. This is one of the calmer days I've ever seen there. So this is a place where there's not so much Ascophyllum because normally there are very high waves coming into the shore. But there are plenty of rockweeds here. We have about 5 rockweed species in addition to Ascophyllum in Maine. And so in the high zone, you have Fucus spiralis. The mid zone, you have Fucus fasciculosus. And down in the low zone, you have Fucus disticus. Okay. This is a collection of electron micrographs that came from David Garbury's lab at St. Francis Xavier in Antigonish. And it shows something that David has emphasized, which is that Ascophyllum has the unusual characteristic of essentially being a lichen. You know, we think of lichens as being a photosynthetic plant or green alga, along with a fungus. And in addition to being so very abundant on sheltered shores in the North Atlantic, Ascophyllum's cell walls are penetrated by many fungi, and they belong to a genus called Mycophyceus ascophyllae, which David Garbury classified. And so here you can see the arrows pointing to areas inside this cell wall between the photosynthetic vegetative cells of Ascophyllum, and they're just everywhere. So the biomass of the fungi is pretty significant. Interestingly, Ascophyllum has lived in North America and in Europe since about 131,000 years ago. In other words, through the last 2 glacial cycles, even though the ice sheets were substantial, the Ascophyllum was able to move to refuge areas, which we think were here in North America and were here by haplotype analysis in Europe. So this is the Eurasian ice sheet, this is the Greenland ice sheet, and then, of course, the North American ice sheet. The current range of Ascophyllum in the western North Atlantic is from Cape May and Lewis, Delaware, which is right across from Cape May, way up into the area close to Greenland. And in fact, Nuuk, which must be about here, has a little bit of Ascophyllum. And we'll probably have more because the prediction is, of course, that the warming temperatures are going to cause Ascophyllum to be lost in more southerly latitudes, but to extend farther up into the Arctic. At one point in time, it was thought that Ascophyllum was recolonized after the last ice age from Europe to North America, but in fact, the haplotype, the molecular analysis of both nuclear genes and mitochondrial genes show that they were there in place. Now, if you have a favorite alga and you want to know more about it, what you should do is to put algaebase.org into your search, and about here, a little box will appear, and if you put it in, you will generate a page, something like this. And you can see that there is a classification, and again, the Fucaceae are the rockweeds. So it's not just Ascophyllum that is rockweed. Something I wanted to point out here to you, I think. Yes, the plant generally regenerates new fronds from the base when one of the larger fronds is damaged, so that the stands you see on the shore may be very persistent. Okay. So this is a different kind of way of looking at relationships among different eukaryotes. A eukaryote is an organism in which all of the cells have a DNA inside a nucleus in each cell. And this is an unrooted grouping. And so somewhere in here, there is an origin of the last common eukaryote. But what we have here are supergroups. And so the important thing is that Ascophyllum nodosum, a brown alga, belongs to the Stromanopyle supergroup. And neither Ascophyllum nor any other brown alga is a plant. Plants are placed in a supergroup called Archaeoplastida. And you can see that there are algae here that you could call plants, like red algae and green algae. And of course, the green algae evolved into this little shoot called land plants. Every place where you see pink, there has been something called a secondary or serial endosymbiosis involving a red alga. So the red algae have a disproportionate impact on many of these groups from that process. What I want to do now is to show you something about a very interesting experiment that Bruce Mingy and Jane Lubchenco started in 1974 at Nahant. And when they were there, they requantified transects that they made. So this area started off just covered by Ascophyllum. And then they burned and scraped a 2-meter wide strip, and it went from the low zone and intertidal zone to the high zone. So this shows how well they cleaned the rock from what had been like this. And you can see that when they came back, Bruce Mingy, who just happened to be in the Boston area, and he went out to see what it was like in 2009 and was sort of shocked that the Fucus, which had occupied the cleared area, not recolonization of Ascophyllum here, it was still just like that. And to the edges of the transect, there was Ascophyllum. So Heather Leslie, who's the former director of University of Maine Marine Lab, and Matt Bracken have followed this up in recent years. And this is what happened immediately after the Ascophyllum canopy was removed to encrusting corallines. I can't show you an encrusting coralline here because, of course, they're under things. But if you left the Ascophyllum, you know, they had replicate plots along with the transect, and they had 3 transects. So if you left the Ascophyllum in place, as well as Semibalanus balanoides, which is barnacles, then the encrusting coralline was alive. But if you took the Ascophyllum away, even though you left the barnacles, then in just about a day, all of the encrusting coralline died. And there were effects on some other organisms too, but it was particularly dramatic in the encrusting corallines, which is why they used that as the graph. And then without the Ascophyllum and without the barnacles, things didn't go very well for the crust either. So this demonstrates the fact that one of the important ecological roles of Ascophyllum is to be a canopy that protects subtending organisms, whether they are attached or mobile, from high temperatures and desiccation and high light. So Ascophyllum is a foundation species in the North Atlantic intertidal zone. It is subject to very heavy grazing by incredibly large limpets in parts of the northeastern Atlantic shore. And so you can have rocks that almost look like this because the patella has grazed off all the algae. But fortunately, we don't have any of those. Now, so these terms are very good: canopy, foundation species. On very rare occasions, I will see someone say seaweed forest or Ascophyllum forest. That is not appropriate. Okay, here I am back out at Winter Harbor a few weeks ago. And you can see that there's all this Ascophyllum, which is yellowish. And then you can see a lot of Fucus vesiculosus, especially on edges of rocks. And I wonder if that is in part due to the fact that in the storms we had last year, this area was battered by rocks being thrown up, and the Ascophyllum was removed and Fucus expanded. This is Fucus vesiculosus too, farther down. Now, this demonstrates the fact that Ascophyllum really needs more shelter compared to Fucus vesiculosus. So here we are looking out from Winter Harbor directly into Frenchman's Bay. And you can see that there is some buffering of wave force of waves that come in here by these blocks that we can see and others like it that are below water at that point. And so notice that you can actually see the boundary between the Fucus vesiculosus and the Ascophyllum. So if Ascophyllum were growing here, it would be broken at a higher rate and Fucus can get in underneath it. Steve, are you on?
Yes, I am.
Okay, do you want to tell us about these two images? I've got the one that has lots of algae, including fucoids.
Yeah.
This one.
So this picture was taken by my collaborator, Peter Petratus. This is on Swans Island, Maine, and it's at the Mill Pond in Burnt Coat Harbor. And this picture was taken about 1983, and it shows the whole shoreline around Mill Pond being dominated by mussels and barnacles. And the locals that live on the island have told us that site had looked that way since the 1950s. So for the better part of half a century or more, it had always been dominated by mussels and barnacles. And then in about 2006, we began to notice that a lot of mussels, adult mussels, were dying. And most importantly, we observed that we weren't seeing a lot of new baby mussels forming in, settling and coming in behind them to replace them. And over the years, it just became fewer and fewer live mussels and a lot of mussel shells that from mussels that died for one cause or another. And we'd been recording the annual recruitment of mussels on this shore and other shores around Swans Island every year. And about 2004 was the last year that we documented recruitment of mussels that could sustain the population, given what we knew the mortality rates of mussels were on that shore. And since 2004, it had been declining at about 15% per year up to the present. And so then several years later, next slide, Susan. This is 2015 from the same spot, the same vantage point, and now that whole shoreline is occupied by Fucus and Ascophyllum. And there are very few mussels at all. Lots of barnacles that facilitate those fucoids coming in, but it is now an entirely Ascophyllum and Fucus dominated shoreline.
Are there mussel larvae in the water?
That is something we have not measured, and I've wanted to. What people tell me is that there are people that do aquaculture find mussels on ropes. So there are mussel larvae in the water column, and I actually was interested in seeing whether in this time in 2004 to 2012 in particular, there were so many lobster buoys and lines. I was wondering if aquaculture people are seeing them on ropes, I wonder if those larvae are settling onto ropes. But then as the trap gets hauled in every couple of days, they just get crushed, and that might be a source of loss of mussels. So they are in the water column. There may be reduced numbers. Whether that is we've correlated the decline of mussel larvae in the water column with greater temperatures in August when the larvae are in the water column, whether it's directly caused by temperature or if it's a correlate for something like food supply of phytoplankton that they're feeding on in the water column, I don't know. And so we don't have a good handle yet on what's happening in the water column.
These are spectacular photos. Thanks so much for sharing them.
Thank you very much.
Okay, so Ascophyllum is fragile compared to Fucus, but it can regenerate by at least 3 different mechanisms. And this guy, Bardseth, was recruited by the Irish Fishery and the Institute for Industrial Research and Standards, but essentially the people who manage fisheries, because he was Norwegian and capable, and they wanted someone who was not Irish to come in and evaluate the rockweed harvest that was really starting to be more organized. So he found that, well, he did a lot of experiments, and you can see that because here's the holdfast of an Ascophyllum. And then the top part is cut off. And 5 months later, this is what that individual looks like. So it wasn't cut below the holdfast. This is where it would have been on the rock proper. And so what you can see is that there are a lot of these base shoots. So here's a base shoot, and 5 months later, it has grown out quite a bit. And Ascophyllum has dichotomous to irregular branching. And so you can see that this is a dichotomy starting. And you can also see that the stumps also have lots of small new branches that are forming. And these actually happen through in vitro embryogenesis. I've done this myself. And so what you see is that a little bit of the tissue close to the surface reorganizes, and under a dissecting scope, it looks just like a small embryo that would have been created from fertilization. And of course, this sort of thing is fairly common in plants as well. So the base shoots at the holdfast can grow into be new axes. The uppermost lateral branch can restore apical dominance and become the leading axis. And if damage is really severe, you will have a lot of in vitro embryogenesis on both small fragments of holdfast that are still going to be in the substratum. Now you're going to have to turn on your side. I know, I apologize. But these are the base shoots that you were just seeing. I mean, not the same one, but here's one that would have been formed from the holdfast proper in August. And by December, it has a number of branches, and also it has some more new basal shoots of its own. By March, it has formed receptacles, which will contain either sperm or eggs, depending upon whether this is a male or a female. And then this is now a year later, and you can see that it's much better developed, and there's a lateral shoot growing out. And then this is still a year later. So within 3 years, you've got essentially a restoration of a normal looking individual. Now, Ascophyllum nodosum drifts very widely at sea after it's been broken. And you have probably seen this on a nice ferry over to Monhegan or, someplace like that after big storms, especially in the spring through the fall when the receptacles are still on for a while and making it very heavy and therefore or subject to breaking. We don't have ice very much on the coast anymore, but ice, I can remember it at Schoodic when almost a fifth of a mile of the shore at Schoodic had an ice shear, and it took off all the Ascophyllum and all the Fucus. And within a few years, it had grown back in. Now, there was a nice study done by Bob Vadas and Wes Wright and Brian Beal in Cobscook Bay, and they used 5 sites, and they wanted to know how much of the standing biomass in the fall was removed by storms and ice cover due to harsh conditions. And so they found that over the 5 sites, there was variability between 29% and 71% of the standing biomass being removed. But when they pulled the 5 sites for a mean, it was 54%. So it's clear that there's a lot of Ascophyllum that is being removed, and a lot of it floats offshore, goes into a detrital pool, and so forth. And this particular specimen is in the University of Maine herbarium, and it was collected near Cape Hatteras, North Carolina, floating in December 1967. At that point in time, there would have been some Ascophyllum on the Maryland shore, but Ascophyllum from North America is found drifting out towards Bermuda and, you know, all sorts of places. So it's a great dispersal mechanism in itself. It never becomes reattached. It looks sort of like a scorpion. It's called Formis scorpioides, in fact. Now, Ascophyllum has been harvested and put into soils in places like Scotland and Norway and Ireland and Wales for at least a period since the 1600s, which probably means much longer before that. But more recently, the agricultural benefit of Ascophyllum as a sustainably harvested source of nutrients and biostimulants has become more clear. So many studies have found that Ascophyllum nodosum extract, and this is actually a commercially available compound. In other words, this is something you would buy A&E. Can protect against freezing or drought in model plants like Arabidopsis, and also in agricultural crops and trees. And their favorable effects on plant roots in terms of increasing branching, increasing leaf area, etc., of A&E treatment by foliar spray or drenching the soil with a low concentration of A&E and water. And it's evident across multiple species and numerous well-replicated experiments, and a good review to look at for this is this. And although the benefits of Ascophyllum in agriculture have been long known, we're fortunate now to know why. And that's because of the molecular techniques we have to look at what genes are being expressed in response to a particular treatment. And so this is some work that Hines et al. Did. And they found that Ascophyllum nodosum stimulates the arbuscular mycorrhizal fungi and their endomycorrhization of plant roots in fields. So, you know, we all sort of know now from popular books, as well as, you know, our own reading, I guess, that the soil is a pretty active place, and that there are fungi making connections between all sorts of higher plants. And so this extract helps to have that happen earlier. And so so these are week blocks, whether the effect was in week 1 or week 4 or week 7. And here is the treatment block for control. And the control was just the analyzed minerals that were extracted from the Ascophyllum, and in the dilution were at micromolar quantities as shown here. Drench is this sort of dark area, and darker is a foliar spray. And the results were not always the same in terms of how you applied it. And different plants had different responses to either drench or foliar. And here are the codes for the genes that were followed. And this is something called a heat map. And so it's also on a log scale for expression. And the brighter colors mean that there is a higher effect. You know, there's more effect of the treatment. And so if we just look at some of the genes, PT4 is a phosphate transporter localized in the periarbuscular membrane involved in acquisition of phosphate released by AMF. RAM2 is a glycerol phosphate acyl transferase involved in lipid biosynthesis that enhances hypopodia formation. And where's endo? Endo has a little bit of a, I mean, you know, this is an effect down here. It's just not an effect like these. But endo, where is it again? Putative cell wall repetitive hydroxyproline-rich protein, often used as a marker of early symbiosis establishment. And so it's no longer as if we're putting snake oil on something, not that people thought it was snake oil, because over centuries they had observed the difference it made in their agriculture. But it's quite important to know what the effects actually are. And so I had originally intended to put in more of the papers, but I have come to sort of the end of the time I'm supposed to take. And so anything you want to discuss or ask about now would be great. And I think the idea of alternate stable states is less talked about now, but it has been a force in ecology for a while. And so at one point, people thought that Ascophyllum and Middleus were alternate stable states. And that's a little bit of what Steve was showing at Swan's Island from the old photos. Or is the alternate stable state Ascophyllum versus Fucus, as we have seen in the Mingy, Levchenko, Bracken, Leslie experiment at Nahant, Mass, on those transects, in which Fucus is still stable there after the removal of the Ascophyllum? Or is the outcome of these comparisons sensitive to climate change because air and sea temperatures are rising and Ascophyllum is receding a little bit, especially in the northwestern Atlantic? The southernmost place in the northeastern Atlantic is Viana do Castelo, which is one of my field sites in Portugal. And so I haven't been there for about 5 years, but you wouldn't call the Ascophyllum abundant, but there was still Ascophyllum there then. And there's been another paper that I saw recently, just published 2 years ago, saying there's still Ascophyllum. And I think it would be interesting to think about how our understanding of rockweed biology and ecology affect our understanding of rationale for DMR's rules for harvest of Ascophyllum. In some places in the world, Ascophyllum can virtually be clear-cut. That is not true in Maine. In Maine, the lowest cutting height allowed for harvesters with permits from DMR is 16 inches above the substratum, with the branches of the main axis below that minimum cutting height left intact. Some of the cutting is done by hand with rakes, although I think a lot of that actually still occurs on a boat. And there are also mechanical harvesters that are used off boats by several companies. And inevitably, because of the way the boat rises and falls and has to be above 16 inches, the real achieved cutting height is very wavy, and I don't know what the minimum is. Maybe somebody on the call would, if some of the harvesting companies have joined us. But I would bet that it's rarely below 20 inches and maybe higher than that. So I would be very pleased in the spring to lead some field trips on the shore, and I could do it in 2 different places on the Maine coast, given the spread of people relative to where you live. And I could also pick up the other half of my presentation, which I cut when it got too long, to tell you about the early life stages of the rockweeds and things like that. So thank you very much for coming. And that's it.
Great, thank you. So I have tons of questions, but I'll hold off. So if anybody has some questions, go ahead and raise your hands and I will call an order. Or I can give the book. Oh yes, go ahead, Helga.
That was great, Susan. I really enjoyed that. Yeah. There were a couple aspects that I was amazed about. First off, that Ascophyllums were like a lichen than I'd ever thought. And the other that sort of goes with that is that it's not a plant. I mean, the definition of plant is obviously a question we can discuss, but the lichen, so what are the benefits and do you know much about that relationship between the fungus and the algae?
David Garbury has published some very interesting papers over the last 15 years. And in one, the fungus has to infect after there is a new Ascophyllum, baby Ascophyllum. And so in the lab, he was able to test stress tolerance with Ascophyllum that had no fungi, and Ascophyllum that had been inoculated, so they had symbiont fungi. And the inoculation with the Mycospherius led to greater stress tolerance.
Wow, that's really interesting. Yeah.
I could just add a quick question. I'm John. You haven't talked about, I think they're feeding Ascophyllum to cattle or something to reduce methane production?
Well, not to produce methane.
To reduce methane production.
There is an red alga called Asparagopsis.
Oh, okay.
When fed to cows, can reduce methane production.
Okay.
I don't know of any place where it's being done at scale, although I think the Bigelow Lab has had a project with dairy farmers in Maine, and I don't know what the results have been, whether it's promising or not, and what else might be involved. Asparagopsis has some nasty compounds in it, but that doesn't mean they get transferred into the cow. So actually, there's an interesting story about this because the Maine Seaweed Council was founded in 1978. Is that right? Anybody who's on who will know the date? Was it 1978?
That sounds about right.
Okay. And by an incredibly large group of conservationists. And so every time the Maine Seaweed Council meets, we read a pledge about taking care of Maine's ecosystems with rockweeds before we start the meeting, and other things too. But Susan DeMisi, who unfortunately died a year ago, was really a leading light in making sure that conservation was at the heart of the Maine Seaweed Council's work. And she was interested in racehorses. And as a relatively young woman, she had noticed that her racehorses seemed to be having some problems that made her suspect that they weren't getting enough minerals. And so she was, very, what should I say? Yeah. And so she had it analyzed, and she had Ascophyllum analyzed, and she realized that if some Ascophyllum were put into the feed for horses, it would probably get rid of the problems they were having with their feet and bones and so forth. And so she did. And I think most of the horses that run in the Kentucky Derby have been eating 5% or so Ascophyllum in their feeds for a long time.
Yeah, that's fascinating.
Yeah. But you can't go over 5%, I think, without problems occurring,
I can add something to that. George Seaver, I knew Susan since the beginning. It was about a coffee scoop per day for a large animal of her source product, it was called. It was a blend of rockweed materials. I think the original rationale for the sea plants, sorry, seaweed, contained these minerals. I think ultimately, and it also relates to the dairy question, I think what she ultimately concluded was that it just improved the digestion, the gut health of horses or cows or sheep or dogs, so that whatever nutrients they were getting, they were able to make better use of. And that also fit into the idea of reducing methane. If you have better digestion, you have less methane.
Mm-hmm.
So do you think that was the alginate from the cell walls that was doing that, or something else?
I don't know. Just something about it. And again, you know that I do biostimulants. There's lots of mystery. In fact, I'd love to ask you offline more about the possibility that it's the fungus that promotes the growth more than the seaweed. But that's another question.
Yeah. I think, based on what we know about this fungus, and David's one paper I know in which he could demonstrate that infection with it created stress tolerance.
Yeah, makes complete sense.
Mhm.
But incidentally, our harvesting for the amount we need for our biostimulant business is all hand cut with machetes at low tide with boots on, and you carry it up the beach in trays. So, and they go back to the same beds every couple of years, or if they're light on that bed, they go back the same year. And they're cutting the big plants before Mother Nature swipes them.
Right. It will be very hard for us to stop calling it a plant.
We process it at our plant.
Yeah, exactly.
All right, so sorry about that. My puppy just knocked over everything. So if you heard commotion or anything, I do apologize. So Dan, you have your hand raised. And so if you'd like to go ahead and ask your question.
Oh, I have a question. This is David Porter. I'm in London. I'm a trained mycologist, and it's so nice to hear all of the discussion about the fungus that's symbiont in Ascophyllum, because those of us who've known about this, like David Garbry and others, for many, many years, really feel we've been hitting our head against a stone wall trying to expose this very interesting relationship. And it's nice to hear you talk about it right up front, Susan. Thank you for doing that. And I don't think that the jury has come to any conclusion on the value of the fungus for the seaweed, but it's really interesting that it's one of the very few fucoids and very few brown algae that has an endophytic fungus in the in its tissue. So it's really quite interesting from that point of view. And we just need a little more research to sort of scope out what's going on in the relationship. I do have a question for you though, and that is, I wonder if there has been any measure of the difference between the drift seaweed that we see out in the bays and the amount that has settled out as what people are likely to call blue carbon additions, compared to that which is washed up on the shore, which is unlikely to be considered blue carbon as far as its function in burying carbon in the land. I don't know that Vadas correlated those two or did any studies comparing the drift versus the washed up seaweed. Do you know?
I don't think so. And both are such a large biomass, and it would be an interesting undertaking, I suppose. But I'm very skeptical of any brown algae being carbon sequestering. Yes,
I agree with you there.
They fix incredible amounts of CO2. But sequestration is a different matter.
Yeah.
They are just covered with bacteria. And I've been analyzing the microbiomes on a lot of Fucus species, as well as Porphyra, which is a red alga. And we've done the genomes of a number of them. And they have so many polysaccharide digesting enzymes. So the likelihood of anything really sinking and providing a meaningful amount of carbon out of circulation seems unlikely to me. Not to mention, of course, that we need to be worried about deep sea communities.
Right.
Okay, thank you. So Kate, you have a question, Kate Jackson. Would you like to come off mute and ask your question, or would you like me to read it?
I get it.
Yeah, we'll read it.
Got it. Got it.
Yeah, go ahead. Sorry, no, I know it takes a second, so I wasn't rushing, but go ahead.
So, yeah, I missed the first 10 minutes or so, and so maybe you already covered this, but is it important for the ecosystem function to try and restore more of the rockweed?
I would say that the Ascophyllum does a pretty good job of that, with the possible exception of places in which the competition with Fucus is limiting space for Ascophyllum. And that was the point that I hope to generate with the discussion point 2 I put up. So in places where people really clear-cut Ascophyllum, they expose that area to recolonization by Fucus disproportionately. And that would be bad for, the Ascophyllum and the various things that depend on it. But Ascophyllum regenerates so well. It's really just a champion regenerator. And there are many studies that have shown that, lots of photographs on the Maine shore with just a difference of a few years, and you see the canopy reappearing. So I don't think there's a need for anyone to be worried about starting a restoration. I mean, as you saw from some of the photos that have been shown, Steve's as well as mine today, there's a lot of Ascophyllum. And I had a brief note from Walter 80 just before we started. Do any of you know Walter 80? He was at the Smithsonian for a long time. He's coralline algal expert. And he's still working at the age of 91. And he was going to come tonight, but the proofs for his latest book have arrived with the index all screwed up. So what he's doing is with an assistant. He's fixing the index before he has to have it in tomorrow. So at any rate, he wanted me to tell you something. which was that Ascophyllum's primary productivity is huge and much more important than kelps. And he has been an expert in the North Atlantic for a long time. So I think that is a reliable sentiment.
You bring up a good point, and that is, or he does, and that is that the focus that we've had on subtidal kelp has seemed to be overblown compared to the need to be focusing on the Ascophyllum and the intertidal. Would you agree?
I think it's important to think about them both. Again, I do not think the rockweed is threatened in any way. That's my scientific judgment. But I also, they're different issues of kelps. I think kelp aquaculture is good. I wish we were not putting so many eggs in one basket by doing Saccharina latissima to the exclusion of others. And I and Sarah Redmond, who is a co-founder of Springtide Seaweed, developed Alaria esculenta, which is in a different family of the kelps from the Saccharines. It is closely related to wakame, which is the huge food kelp in Japan. And, there are many ways in which you can use sugar kelp, Saccharina latissima, but I don't really think it tastes good. And wakame and Alaria really taste good. And so I can't quite understand why someone in the state government doesn't try to create more of a focus, since we know how to do it, to expand the Alaria aquaculture. The other thing that concerns me is making sure that fouling organisms are removed from aquaculture gear at the end of the season and don't, perpetuate the spread of invasive species and things like that. But that's a very manageable problem. It just needs to be addressed.
Can I just make a comment?
Well, can I, Dan had his hand raised. Sorry to interrupt everybody. Dan had his hand raised about 15, 20 minutes ago. And I just want to make sure that he is able to ask his question.
No, please, on the train of thought, keeping the train of thought. I can be patient.
Okay, go ahead then. Sorry, I just want to make sure I got you in. So
I don't have the little hand on the bottom of my screen. Anyway, I heard the word clear cut a few minutes ago, and that of course made what's left of my hair stand on end. No commercial harvester of Ascophyllum would be so stupid as to clear cut, because they want to go back to the same beds soon. So it would be an unthinkable amount of work and time very poorly spent to do anything but harvest it per the rules. It's not in the interest of anybody who's commercially harvesting it to harvest it too short. I mean, you'd just be crazy, and it's a lot of work if you tried to. I don't know how they did the research. Well, I guess they must have had razor knives or something. But at any rate, there's nobody commercially harvesting who would ever even remotely consider clear cutting, just because you'd be crazy to try. Susan made the mistake of using the phrase clear cut, and it got my blood going.
I wasn't referring to anybody in Maine.
I know you weren't, but
in fact, the point I was making was that from what we know of the dynamics between Ascophyllum and Fucus, it reinforces the standards that Maine adopted to protect the Ascophyllum and make sure it regenerates well.
With respect to the sinking for carbon credits or the going up on the beach, I spend a little bit of time on a boat every summer, sailboat, and there's lots of floating mats of rockweed out there. You have to shut the motor off or you suck them into your cooling system. My sense is, and I don't have any way to document this, but intuitively, and I base this partly on how a few years ago the wind was blowing a certain way, and Rockport Harbor had like a foot thick layer of Ascophyllum floating in the whole harbor because it all blew in there. I think that it all ends up on the beach ultimately because it's very slow to sink. Those little bladders keep it up for a long time.
Yeah. Halfway across the Atlantic in many cases.
There you go.
Well, you know, up on the beach, it's still very valuable food for a lot of organisms.
Right.
But too much on the beach creates a big dollar problem for resorts and things like that. So I guess they need to deal with it a bit.
I'm glad we don't have sargassum up here.
Gee, I was just thinking of it.
Yeah, Dan, if you wanted to ask your question.
Yeah, so on that topic, have there been instances where overharvesting has been demonstrated to be consequential to the recovery of the Ascophyllum?
Not that I know of. I mean, I'm only speaking for our area, right? And so I don't know whether something has happened in another place, but not here.
Because the studies with the clearing show that there's recovery. And for example, the instance where uh, the muscle hysteresis uh, resulted in Ascophyllum coverage. So I was wondering if there is, dispersal limitations that could potentially result in consequences given a large enough scale of overharvesting, or if there were any, if there would be any consequences by changing those types of regulations to allow for more harvesting. What do you think about those?
Well, you've posed a lot of different questions. I think that one of the issues is what the recruitment of Ascophyllum is. And some of the slides, I cut out a lot of my own slides, but I studied Ascophyllum recruitment just north of Bucksport on the Penobscot River. And the reason was that I'm very interested in reproduction under low salinity habitats. And it's taken me pretty much all over the world from the Baltic to low salinity habitats and estuaries. And the Ascophyllum released a lot of gametes, and the zygotes stuck to the substrata that I put down on the shore. And, there were a lot of recruits. But in some of the literature, people don't find recruits. I don't know whether they get eaten quickly, and that reduces what people are seeing, or what. That's probably an area that would be interesting to investigate in different parts of the shore.
Is the fungus, is that carried from the parent via the zygote?
No, it has to be reinfected from open water.
So I'm at the Woods Hole Oceanographic Institution, and I culture seaweeds, and I'm right now I'm doing aquaculture, but I come from I did my master's in ecology, and I'm looking for a direction to study Ascophyllum, and this seems like an interesting one to expose the fungus in isolation, two treatments, exposed and non-exposed, and see the difference in development. But do you, in your opinion, do you think there's other low-hanging fruit for culturing studies, helping elucidate any sort of Ascophyllum ecology?
I would suggest you read David Garbury's papers, because he's written like 7 or 8 of them, and some, they're in different areas, but it would give you a feeling about this fungus. There is so much fungal biomass inside Ascophyllum, it's amazing, because all of the cell walls are so heavily penetrated. And those spores have to land on the ascophyllum and then penetrate into the cell wall and start to grow between the cells in the cell wall. But, you know, a conversation with David Garbry might be very helpful to you. I'm not absolutely certain he's still teaching full-time, but he is on the website still, and I'm sure he'd be really delighted to talk with you.
Thank you. I have one more. You listed a whole bunch of characteristics about Ascophyllum that would sort of allow for it to become the competitive dominant. But would you say that any particular characteristics is overwhelmingly contributes to that dominance?
Is Steve still on the call?
Yeah, I am.
Can you answer that?
I think longevity. I think because we know that, as Susan was talking earlier about recruitment of Ascophyllum, and we measured recruitment for decades annually, and the mortality rate of zygotes and germlings is, it's over 99% per year. And I think it's not so much, I think more being removed by waves and drying and probably high temperature is more important than grazing. Grazing happens, but I found it to be very episodic and variable. But it doesn't matter for a seaweed that lives for decades, scores of years to maybe even a century. Even if it takes them 13 to 15 years to replace themselves, they've got a lot of opportunity because they're putting out, the estimates are like 2.5 billion eggs per meter squared per year. And we think it takes about 10 to 15 years to replace oneself. Once a zygote settles and establishes, if it survives, it'll be about a dozen years before it's likely to replace itself. And so it's a lot of time with a lot of zygotes, and they don't have to reproduce that frequently because they live for so long, and they really are tolerant of environmental conditions that other species are not, and they kind of dominate what happens on the shore.
I like the longevity approach, but I'm thinking, and here's my perspective. I'm coming from California. I actually just moved to Massachusetts 2 years ago, and I studied kelp on the West Coast. And there's the subtidal community has a number of different kelp species, and there are some stipitate kelps over there with harder woody stipes that grow in the seafloor, and they live for very long, partially because they can store nitrogen in their tissues and survive and live longer because of that. But there is the giant kelp, which is the competitive dominant over there, and it doesn't live as long. So it's the longevity definitely helps them persist when they aren't outcompeted by the giant kelp. But just my perspective, and I'm really new to the coast. I mean, 2 years is still new, but it's just so amazing that they can persist. And I guess I'm sort of projecting here. This is actually my personal experience. Persist in a different climate that is so seasonal compared to what like kelps, the West Coast kelps would be exposed to.
Well, they are quite different algae, but I think that the fact that Ascophyllum is so uniquely capable of regeneration is one of the most important things to remember about it. I think it swamps out recruitment by new zygotes in most cases. And you only need to have a tiny, I mean, like a square millimeter patch of residual holdfasts. I have seen regenerate into canopy of Ascophyllum along the shore of Schoodic, where it was completely removed by an ice shear about 15 years ago. And Tony Chapman and one of his students, who I'm forgetting his name, but he's in Australia now and doing great work, but they documented something very similar along the Nova Scotian shore. So it's great that the zygotes, you know, are made and are established. I'm sure that's very important too. But it's the regenerative capacity that is just extraordinary about Ascophyllum. And the fact that those basal shoots are suppressed until the canopy opens, either because of storm breakage or harvest or whatever.
So we are at 8. If everybody still wants to stay on, we can stay on for about 15 more minutes or so. This is good conversation. I still have plenty more questions, but if people want to stay on, then we can stay on. Helga, I still see your hand is up. Is that still up?
Yeah, both of us have questions.
Oh, well, my hand.
But it's up to the host. It's up to Susan if we're willing to stay on for about 15 more minutes.
Sure.
Yeah, okay, go ahead then.
I remember once a long time ago, 67 or something, in Newfoundland, seeing detached Ascophyllum growing in tight little balls in very protected areas, up in backwaters somewheres. And I think it was like just basal shoots. There were no receptacles or air bladders or anything like that. And they'd form little tufts of 6 to 8 inches and a little bit flattened. Have you seen that?
There is, in fact, another form of Ascophyllum. And that was one of the last photos I took out. Let me just see if I can find it real fast and I could show it to you. But yes, there is a salt marsh form of Ascophyllum, which is very minute and heavily branched. It doesn't attach most of the time, and it's quite abundant in certain habitats.
Okay.
I don't know whether I'll send you the slide, John.
Okay. Sure.
And I was wondering,
It's called Mackii, so I think it's named after a Nova Scotian Scot.
Okay.
I was wondering, you know a lot about Ascophyllum, but you also got Fucus some of the Fucus species that can make a really big, wonderful canopy. And they also would probably get damaged and need to regenerate. So what are the comparative regeneration capacities of the other fucoids relative to Ascophyllum?
You're correct that ice shear would treat them equally, but not storm breakage. Ascophyllum is quite fragile. You know, it's like it's got this relatively slender axis and branches coming out that can get tangled and caught. I mean, it doesn't seem like it would be very resilient when you just look at it. And in fact, it's not. It breaks very easily. Whereas Fucus is basically a planar type of body construction. And so you can imagine it going back and forth with waves. Oh, I found it. Now, how can I show this?
You have to share your screen again.
Well, yeah, I'm trying to figure out where the screen share is here. Can you see this?
No, not yet.
Having lost the share, I wonder where I find it. I'm not getting the normal kind of menu option on my screen that I expect. Well, that is a shame. Maybe if I hit this button.
You are a presenter, so it should be able to share.
Is it a pull
Corinne,
down
could you
to
have
start
any
share
input?
under meeting?
I see a little green arrow at the bottom next to the reacts that says
Oh, yeah,
share.
that's there
Yeah,
too.
right
Yeah,
there.
that's there.
You can see that?
Not yet.
It's there.
Okay, well, I hit that arrow. Okay. Now I think I can get it.
Aha.
Ah,
Yes.
oh,
So you
yeah.
see Ascophyllum nodosum formamicai. And I think that's it's probably Mackay. I've never heard it pronounced. But I think this must be named after a Canadian. That's my hunch. I think there was a Mackay who would have been a candidate for that. But you see, it's very finely branched. It doesn't attach. It doesn't form receptacles, but it does grow. Whereas the Scorpoides that I showed you, which kind of grows in crazy ways as it gets into the drift, isn't nearly as pretty as this, right? This has a pleasing architectural form, even though it's detached.
That's probably it, yeah. That's interesting. Good.
Yeah.
Oh, you were comparing Fucus and
Yeah, so you didn't really talk about the regenerative capacities of Fucus relative to Ascophyllum. You said
I
that
don't-
Fucus would
I
be
don't-
as
Yeah.
bothered by, it would be ice shear, but not storm damage.
Well, it does come back from ice shear, so it's the same thing. I mean, Fucus will regenerate from bits of its holdfast that are left. In Tony Chapman's study, I think that there was very little left of a holdfast, but it was enough so that, again, it reorganized, and under a stereo microscope, you would see little dome-shaped things that look like embryos, except they're, of course, still attached to the remnant of a holdfast. So it certainly regenerates. But it doesn't tend to break in the same way that Ascophyllum does in the abundance. It would take a lot more force to do it. And of course, sometimes that happens, sure gets cleared.
I really loved your diagram showing these different shoots, and then, you know, they'd sort of be waiting for light, just like in a forest when there's an opening, then all these little plants start growing faster. And it was really the comparison, the canopy in the sea versus the canopy on land. This is really, really good images. I really appreciated that.
Yes, just remember, it is not a seaweed forest.
Oh, I know.
Well, you can probably call kelps that grow to 30 meters a forest if you want to, because it becomes a somewhat more, it's not the same, of course, but the way in which it's reacting in the community is more like a big tree. But it is quite remarkable, the ability of the organisms to provide, I mean, Ascophyllum and Fucus provide protective canopy for a whole host of invertebrates and other algae that are underneath that would get blitzed by high light or high temperature, or if they're low, too much drying without that.
Well, Susan has a question. If you want to go ahead and ask, and then we can start wrapping things up if people do not mind.
Okay. Hi, Dr. Brawley. This is Susan Burnett from Portland. Thanks for being here tonight. And I just wanted to ask, I was just curious, but I may have answered my own question. The Mackay doesn't attach. So at first I was thinking, well, then how does it stay in a particular area? But then I realized, oh, if it's in a salt marsh, perhaps there's not that much movement. So maybe that's the reason.
Right. That is the reason.
Thank you.
And so it's growing in the marsh grasses, right? So that retarding the water movement so that it can just sort of sit on the sandy, muddy surface and grow in that somewhat pleasing and structured way, even without attaching.
Thank you.
Thank you.
Okay, I see Grace Quinn has a question. This will be the last one. So go ahead, Grace.
Hi. Sorry to sneak one more in before. I'm sure we could talk all night. Thank you for such a great talk, Dr. Brawley. And not to bring things back to the endophytic fungus so often, but I know that there's kind of a frequent epidermal shedding to remove epiphytic overgrowth. And I'm curious if, because obviously there's some cell wall restructuring there, if there's any impact on the endophytic fungus that you know of, or if I just need to do some reading of Dr. David Garbury's work to learn more about that.
My impression is that the cell walls that are most heavily penetrated by the fungus are below the epidermis and the myristoderm cells, which means that they are below where that cell wall separation occurs and the epiphytes peel off. I mean, I'm sure some must be lost, but, you know, relatively speaking, the fungi are deeper in the thallus. You know, algal bodies like Fucus and Ascophyllum and Saccharina and whatever, their cell body is called a thallus, Thallus. And so that's one of the things those electron micrographs showed, that, you know, you had a lot of smaller cells with an occasional chloroplast as you go deeper, but it's more sort of structural cells at that point and not the outermost photosynthetic area of the Ascophyllum.
Thank you.
Can everyone see my cat? I'm not sure I'm on screen anymore. I can't see you.
Oh yeah, no, you are. No, can't see. Okay. Well,
You have
thank
a
you so
kitten.
much. Susan, if you wouldn't mind, if you care to share your contact information, this has been a great conversation. I learned some is way over my head. So, but this was great. If you don't mind sharing your contact information, if people want to reach out more.
Yeah, that's fine. Again, I am, since I went hunting, a little bit handicapped by the original Zoom. Okay, now I should have a chat. Yeah, chat.
Great. Well, thank you so much, everybody, for attending. I love it when conversations go over, and we could go on for hours. And I still have my questions to ask, but I will reach out separately, Susan. And again, thank you so much. And I look forward to the next one, which will probably be in January, because we are going to be having a candidate forum with the senatorial candidates. So there will be notices going out about that, and that's going to occupy the next month or two. So thank you again, everybody, and have a wonderful evening. And Professor Brawley, Susan, thank you so much. Thank you so very much.
I really enjoyed everybody's questions and comments
Yeah.
and discussion. Thank you all for coming.
Yep. And when I trek up to Bangor, we'll go to Patty's and listen to music, and I'll ask you my questions. So fantastic. All right. Have a great night, everybody. Thank you so much.
Good night.
Thank you, everybody. Bye.